Yep. All right. Yeah, thanks everybody for joining us. Hopefully there's some more people coming, but I think we can just start now. Yeah, welcome to this month. So like originally it's supposed to be the August JETZON call, but now we're already in September. How does it happen? Yeah, JETZON is the joint, let me think about this, joint exploration of the Ocean Twilight Zone initiative, something like that. I'm really bad with acronyms, but anyway. So basically lots of programs that focus on the Ocean Twilight Zone, biology, carbon flux, all kind of things. And once a month we have this open call where we present, or people present from the community the recent research and new papers and all kind of things that could be interesting. And yeah, so we have one month we have it on European time zone, the European afternoon, and this is the Oceania call, so a bit more friendly for people in Australia who can't get up at 4 a.m. or 3 a.m. at night to give a talk. We have two amazing speakers today, and I think we will start with Michaela Larsson. She's currently based just south of Perth in Western Australia, and I just let you introduce your talk and your topic yourself.
Yeah,
if you just want to share your screen, that would be awesome.
Sure. Okay. Can you all see that? Yeah. And can you see my mouse clicker as well? Yeah. Okay, perfect. Okay, so I just wanted to start by thanking the organisers of this seminar series for inviting me to present this work today. I'm really excited to be presenting to the Twilight Ocean Zone community. I'm really interested to hear your perspective on what you think the fate of mucospheres are. So I'm looking forward to the discussion at the end. So as Svenja mentioned, I'm over in Western Australia. I live down in this southwest region, which is this pink little corner at the bottom here. It's famous for its beaches and its food and wine, but of course we have a jetty as well. It's a 1.86 kilometre jetty, which is the longest jetty in the Southern Hemisphere. So the longest timber pile jetty in the Southern Hemisphere. So we're very proud of that. But I wanted to start by acknowledging and paying my respects to the First Nations peoples and traditional custodians of the land from which I present today, who are the Wadandi Noongar people. But the work I'm presenting, I did when I lived on the other side of the country in New South Wales, when I was a postdoc at the University of Technology in Sydney, a place that I'm sure you all know very well, famous for the Harbour Bridge and the Opera House. But I would also like to acknowledge and pay my respects to the traditional custodians of the land of which UTS stands, who are the Gadigal people of the Eora Nation. So the work was funded by the Australian government in an Australian Research Council grant awarded to Martina Doblin and Sinead Collins, pictured here with me. The work was titled Biological Bet Hedging in a Variable Ocean, and we aim to better understand how phytoplankton will respond to a more variable future ocean. The work that I'm presenting today is also being published in this paper, so I'm going to have to skip over a lot of the methods in this talk for time, but please check out the paper or feel free to send me an email if you would like some more detail. So I wanted to start by defining some useful terms up front, and for reference, I'm using the definitions of the types of plankton that are described in this paper by Aditi Mitra. So phytoplankton is, of course, the microscopic plants. They're unicellular plants that are phototrophic, so they gain their energy from photosynthesizing for growth. And at the other end of the spectrum, you have your zooplankton, which are heterotrophic, so they consume other organisms to gain their energy for growth. Then in the middle here, you have mixoplankton, which are both phototrophic and heterotrophic organisms. So they can photosynthesize like plants, but also consume like zooplankton as well. So it's really important to define this term though, because over the years in the literature, mixoplankton or mixotrophs has been used to mean different things. Sometimes it's used to mean organisms that can uptake and use organic carbon, but this was found not to be very good for distinguishing mixoplankton, because a lot of phytoplankton species can also do this. So in this talk, when I talk about mixotrophs or mixoplankton, I mean those that can photosynthesize and also consume via phagoheterotrophy. So naturally, the obvious question is, how does a unicellular mixotrophic organism consume other organisms? And there are three main mechanisms for this. The first is via phagocytosis, where the predatory cell engulfs a prey cell, like in this first image here, usually through the sulcal groove. The second is via a peduncle, which is an extendable appendage that comes out of the predatory cell, pierces a prey cell, and the intracellular contents are removed. This is also known as myxocytosis or cellular vampirism. And the third is via a pallium, which is a veil that's extruded from the cell, engulfs a prey cell, and digestion occurs outside of the predatory cell. So now, keeping all of that in mind, we'll jump into our study. So the mixoplankton species that we used was freshly isolated from a site located in the continental waters of Sydney, about 30 kilometres southeast of Sydney. And with the help of Gustav Hallegraf, who completed the morphological analysis for us, we were able to identify our species as being within the Porocentrum genus, and the taxonomy aligned with the species Porocentrum balticum. So the main features of this species are that they are very small, about 15 micrometers in length. They are spheroid, but laterally compressed, so not quite a ball, but a little bit squished. And they have two flagella that come out of the top of the cell in this sort of apical region. So we also completed phylogenetic analysis, confirming our original identification that the species was in the Porocentrum genus. And it was also very closely related to the extensively studied species Porocentrum minimum, shown here in red, but most closely related to undefined strains. So there is no type material for Porocentrum balticum, and without sequences to directly compare to, we were unable to confirm if our species were indeed Porocentrum balticum or a new species. So we've just called it Porocentrum CF balticum to acknowledge the similarities, but also recognize that identification wasn't possible. So it's at this point that I want to introduce the avatar of our species, which was created by this artist that I was able to work with during this project, Glyn Gorick. Because the cells are so small, I've used graphics throughout this talk to demonstrate the processes that I'm presenting. And so just for context, this is a light microscope image of the Porocentrum cell, and this is the beautiful graphic that Glyn has created. So look out for these throughout the rest of my talk. So we found Porocentrum balticum feeds via a peduncle, as you can see in the video at the top here. This process can take about 30 minutes, so I've sped up this the process so that you can see the whole thing. So in the images here, you can see the peduncle extruding from the Porocentrum cell and piercing the prey cell, which in this case is a green fluorescent cryptophyte. And then you can see here the resulting green fluorescent food vacuole within the Porocentrum cell. We found that Porocentrum could consume a wide variety of prey types, ranging in size from 3 to 20 microns, but it struggled to consume some of the larger species. It's sort of in the range of 40 to 60 micron, and any of the thecated dinoflagellates as well. We also determined that Porocentrum could consume bacteria, And at the bottom here, I've just got an image showing a fluorescently labeled bacterium, which is this green dot, inside a porocentrum cell. You can just see it in the video too, that the porocentrum cell is very happy now that it's finished consuming its food and it's swimming around very excited. So we also found that porocentrum made these really intricate structures that we termed mucospheres. So after a day of photosynthesizing, and typically in the evening, the cells would start twisting like this and extruding mucus and weaving it into this intricate structure, much like a spider weaving its web. And this process can take several hours. So the porocentrum cell remains within the mucosphere once it is constructed, but it's not trapped within. Along the bottom here, I've just got a series of images that I've taken over several seconds, where in the first there was a mucosphere with a porocentrum cell inside, and then I've flashed it with a very bright light, which it doesn't like, and then it has escaped. So it's quickly swum out of the mucosphere. So it's really important to think of this as more like a spider's web than a bubble that it's trapped inside. So mucospheres are also very good at capturing bacteria. So in this video, I've just started at the top of a mucosphere and I'm zooming down through the Z dimension. So you can see that the bacteria are distributed throughout the entire mucosphere and that the porocentrum cell is within. And then just on the right-hand side here, I've got an image of the same mucosphere as the one in the video, but I've stained the bacteria with a DNA stain cyber green, so you can just see them some more clearly there. So mucospheres are also really great at capturing phytoplankton cells. In this video, I'm zooming in the Z dimension again, and you can just see that a cryptophyte cell has just swum in and it's now becoming entangled in the top of the mucosphere, still trying to struggle free, but eventually it'll settle in there and be immobilized. So mucospheres are also really quite strong and can capture cells much larger than the porocentrum cell that it's produced them. In the top video here, we've got a tetracelmus cell that is clearly trying to struggle free of the mucosphere. And then the little cell is this porocentrum cell waiting for its dinner to calm down while it's slowly dragged along by this poor struggling cell. And then in the bottom video here, you'll see a porocentrum cell swim in really quickly. And that is what the porocentrum cells look like when they're not in a mucosphere. So they swim around very quickly. But then if you look at this one down here, it's created a mucosphere and it's sitting within, which is currently very stretched by this struggling Gambiodiscus cell that is trying to make its way out. So I think these videos really nicely demonstrate how strong a mucosphere is and how effective that they are at trapping prey. We were then interested in understanding which conditions mucospheres are produced, and because phytoplankton cultures or plankton cultures originating from a single cell have large amounts of co-occurring bacteria in the culture, it was important to make them azenic and remove this, because we'd shown that porocentrum could eat bacteria as a food source. So to test true phototrophic conditions, we had to remove the bacteria. So to do this, I enlisted my dear friend and colleague, Dr. Anna Bramucci, who helped me remove the bacteria through a series of antibiotic treatments. And I've just got these figures at the top here to show that we were able to get our culture azenic, as shown by agar plating, some fluorescent microscopy, and some flow cytometry. We also did an experiment to see if the porocentrum cells themselves were affected by the antibiotic treatment, which they weren't, which was really great. We've just got growth curves here, and the blue lines are the porocentrum cells in each of the treatments. And this just shows that the porocentrum cells were still okay, and we could continue with our other experiments. So then I did an experiment where I added individual porocentrum cells to 96 well plates in each well, and observed how many mucospheres were produced under different conditions. So the main thing that I want to emphasize in this figure here is that the most mucospheres were made in the presence of prokaryotic and eukaryotic prey. And then the next highest was in eukaryotic prey only. And then hidden down here at the bottom were the treatments where no prey were added. So no mucospheres were produced in 24 hours when prey was absent. So this really shows that mucospheres are produced for the purpose to capture prey and for consumption. And it's not just an inherent behavior that the cells do each day. It's more like a choice that they make. The other thing that I wanted to point out here in this figure was the importance of light. So this green line here shows that mucospheres were produced in the first few hours in the low light treatment, but then production tapered off after that. So this shows that the energy generated by photosynthesis is really important in the mucosphere production process. So it was during these experiments that I noticed that both bacteria and eukaryotic prey appear to be attracted to mucospheres. So the first image here is a cluster of cryptophytes and bacteria that, if you zoom out through the magnifications in these other images here, you can see that almost all of these cells are clustering around where a mucosphere was, and the well around it is almost entirely empty. So this made us hypothesize that mucospheres may be actively attracting prey. So to test this, with the help of another colleague, Dr. Jean-Baptiste Reyna, we used a microfluidics-based chemotaxis assay. So this device is about the size of a credit card and allows you to place a chemical in the well, which is emitted out of these ports, attracting motile organisms, which become trapped in the well, and then you can enumerate how many organisms are in the well and get an understanding of the chemotactic capacity. So to test the chemotactic capacity of mucospheres, we isolated the mucus from the azenic culture using differential centrifugation, and then purified that extract. And when tested, we found that both prokaryotic and eukaryotic cells were significantly attracted to the mucus extract. So this demonstrates that mucospheres do not passively capture prey, but instead actively attract prey, which can be a really important strategy in the oligotrophic ocean. So we found that all of these behaviors also occur in a daily cycle. So the day starts with the cell photosynthesizing, using sunlight, and then in the evening, with the excess energy and carbon, they produce a mucosphere, which then attracts prokaryotic and eukaryotic cells. And then the porocentrum cell selects one to consume via peduncular consumption. And then once full, it abandons the mucosphere with all of the trapped cells, where it then can divide unimpeded, ready for the next day's cycle. So I didn't have time to go into it in detail in this talk, but we also found that porocentrum could produce sexually as well. And that's just down the side here. So once they abandon the mucosphere, they could also engage in sexual reproduction, ready for the next day's cycle as well. So it was at this point that we were starting to wonder what the wider implications of mucosphere production might be for the ocean, and in particular, the ocean carbon cycle. A process that I'm sure most of the audience in this forum is very familiar with, but in short, the carbon is cycled in the ocean through the biological carbon pump, where phytoplankton use inorganic carbon from the atmosphere to convert it into particulate forms. When they die, these aggregate, sink to the ocean floor, and become sequestered. So we started to think about our porocentrum daily cycle in this process, especially this part where they abandon this mucus laden with all of these surplus prey, and wondered if it could be contributing to this process of aggregation, which I note occurs in the twilight zone. So we were further convinced of the importance of this when we used some commonly used stains for transparent exopolymeric particles, finding positive staining with both Alcyon blue and Kumasi blue, suggesting that the mucus is both polysaccharide and protein rich. And we also found that mucospheres sink. So here is just a series of images over 24 hours taken at the bottom of a well plate, where a mucosphere was made in the sort of water column suspended, and then when abandoned, it sank to the bottom of the well. But then we were starting to think, how much carbon could this process really contribute to the ocean, and is it really important? So we did some rough calculations. And now I'm going to quickly summarize the main parameters of these calculations, but if you do want more detail, please check out the paper or send me an email. So we first determined that our Porocentrum species has a global distribution and occurred in abundances that ranged between 19 and 137 cells per litre, but can reach a maximum of about 3,350. We used a euphotic zone of 60 metres and a mucosphere production rate of 23%, which was measured in low light conditions in the presence of prey. We then measured the amount of carbon in a mucosphere and used this to calculate the amount of carbon produced per metre squared in the ocean per day, using that average and maximum number of Porocentrum cells. But then this increases if you consider the amount of carbon incorporated in the captured prey cells. So for this, we conservatively estimated 52 bacterial cells and 10 phytoplankton cells. And so if you consider the amount of carbon that they're also contributing, you get sort of these sorts of ranges, which is higher than just the mucus on their own. And they're also quite impressive ranges of carbon export contributions when you consider other known carbon contributors, which are appendicularians, zooplankton, fecal pellets, and organic aggregates as well. Then if you scale that up across the entire ocean for a year, you get around 0.02 to 0.15 gigatons of carbon per year, which equates to about 0.1 to 0.7% of estimated export from the euphotic zone. So this demonstrates that the foraging behaviour really is an important process for the carbon cycling in the ocean. Now, I just wanted to finish up by throwing in a bit of a plug for our newly formed SCORE working group. It's called MixoNet. So we're going to continue working on mixoplankton for the next few years. So I encourage you to check out this link if you would like to know more about this project. And I also wanted to finish by just thanking all of the co-authors that I have worked with on this project. It was a really great group and I was really fortunate to be a part of it. And I'll leave it there.
Thank you so much. That was pretty good. I mean, also all the definitions at the start for me as a zooplankton person is pretty good. I could follow, you know, that's great. Yeah, I mean, I find it really interesting because, yeah, we were looking at larvation houses and how much carbon they could export, but it's really hard to calculate that because it depends on how much food is around, you know, how much will stick, how big they are. My God, yeah. So yeah, that's pretty impressive. Nice work. Are there any questions from the audience? Don't be shy. You can also use the chat if you don't want to speak up because you sit in an open plan office. So yeah, during the talk, I was wondering, yeah, if it's really, you know, if it's this daily cycle that in the morning or during the day, there's enough light to photosynthesize so that we could use that to, of course, do that. And then during the evening, or what should we do now? The light is gone, basically, you know, then that I have to feed heterotrophic, you know, heterotrophically, basically. So yeah. Oh yeah. So yeah, but then you answered the question basically that there is this cycle of yeah, carbon export basically. Or what I found interesting is that they sit kind of in this mucosphere and they can get out, but then the plankton they catch can't. Do you think why that is?
Yeah, so that's a really good question. I didn't have time to go into it today, but we were interested. We were interested to know if they actually remained alive in the mucosphere or if they are killed in that process. So we did one experiment where we left a mucosphere that had trapped a bunch of cryptophytes for 24 hours. And then I flashed them with very bright light to see what they would And within about 15 seconds, they had all struggled free of the mucosphere.
Right.
So it's really interesting that they are immobilized and don't choose to leave unless they're obviously scared out of there. But they're not killed, so they're just immobilized, almost choosing to stay in the mucosphere.
Yeah.
And
do you think? Go ahead.
Sorry, I was just going to say we also tested it with bacteria by staining with a live dead stain propydium iodide. And found that basically all of the bacteria remained alive in the mucosphere as well.
Yeah. Do you think they would feed? I mean, some of the heterotrophic ones, at least, or they would feed on the mucosphere as well, while being trapped. Might as well.
Well, I mean, there must be something that's enticing them there, right? And yeah, I mean, we would have loved to have been able to purify what it is that's attracting all these mucosphere, but it's just too hard to get enough of the substance to be able to do that sort of thing. But there must be something that they're lacking in there.
Interesting. Oh, super cool. I think there's some questions in the chat. Abby is asking, are there any plans to measure iron binding ligands from the mucospheres, i.e. polysaccharides? If yes, it sounds like mixotrophs could be playing a significant role in iron availability in the areas of bioavailability in the areas like this in the ocean.
Okay.
So do you want to measure iron binding ligands from the mucospheres as well?
I mean, that would be amazing. So yeah, please get in contact. I will say though that I've now moved on from this postdoc, but the porocentrum culture still exists, and so we can still do some work at some point as well. So yeah, I would love to do anything further like that. So please get in touch.
That's pretty cool. Katie asked, why did you decide to use 60 meters as your export depth?
For the photic zone?
Yes, the export depth, yeah.
Okay, so the 60 meters was what we calculated that there was enough light for a porocentrum cell to produce a mucosphere. And we used that, and now I have to remember, I think it was from the Tara Ocean's light measurements, and we did a measurement to work out what that threshold would be at 60, and then I knew from our experiments that mucospheres were produced under those conditions. So that was used as a representative depth for where we thought mucospheres would be produced. Of course, that would vary. I mean, this is why we're saying it's a very rough calculation.
No, I mean, you have to start somewhere. So, fair enough. Oh, good. George is asking any idea on what the sinking rate might be. The large lavation houses can sink at 800 meters per day, but I'm thinking the mucospheres sink a little slower.
Yeah, we didn't measure that, but I think that's really important. So we kept this study really just describing the behavior and the possibility of it sort of sinking and contributing. But I love that the Twilight Ocean community here are into those sorts of calculations.
100%.
And so, yeah, if anyone is interested in doing those sorts of things or those experiments, yeah, please get in contact, because I think that's the next important step in this study, right? We've shown that it can be produced and they can sink, and it may be important, but what is the fate of it after it's abandoned and sinks?
I would also think, on the way down, there would, more particles sticking onto it, so it would be faster, getting faster and faster with depth. But I mean, that's just an, guesstimate. So, yeah, it's interesting. Yeah, and just wanted to say too, the illustrations are amazing. So yeah, they're pretty good.
That was all Glenn. That had nothing.
Yeah, no, all good. Kudos to him then. Awesome. So I think let me check the time. Yeah, let's move on to